Hi there and welcome to this GCSE revision video for Unit 8 Biology, and what we're going to be looking at today is homeostasis. So homeostasis is basically keeping your body in the very fine balance that keeps you alive and healthy, and it is a process in which your body maintains an internal constant environment. And it's obviously important because things like enzymes need a set temperature, and all your cells in your body can only cope with certain set conditions. It relies on nerves, hormones, and the other organs in your body working together. The main conditions maintained in homeostasis are water content, which if you do separate science that you look at in the next module, body temperature, and blood glucose concentration. And your homeostasis mechanisms involve receptors, which are sense organs, and receptors are effectively a set of cells that are in your sense organs that detect a change in energy levels. You then have some mechanism of control, which is normally your brain responding when your nerves firing as a result. And then you have effector organs, which are, as we'll talk about later on, either muscles or glands, and the muscles always contract or the glands always release hormones. So you need to understand about the nervous system, and that electrical impulses or messages are travelled around specialised cells called neurons, which are adapted by being long, allowing these electrical messages to go through. And a bundle of neurons is what we call a nerve. So you can pretty much move between the word nerve cell and neuron, and you won't get penalised, but do be aware that that's what it means. So you could be asked to label parts of a neuron or a nerve cell, and you should know these things. The general, a nerve, a cell, so it still has a nucleus, it still has a cytoplasm, and it still has a cell membrane, and they have the same functions of those things in normal cells. You might get a picture of this, which is more like a motor neuron, or the second one is a sensory neuron, where the cell's body main bit is in the middle. The bit that the nerve impulse travels down is called the axon, and the bit that receives the messages from other neurons is called the dendrite. So in this case, the message will be going that way down the nerve. And between nerves, because obviously they're not tied together, there are junctions called synapses. And effectively, you need to understand that these work by having an electrical impulse arriving at the end of one nerve. That releases a chemical or a neurotransmitter which diffuses across the synapse, and that activates a nerve impulse in the second nerve, and therefore that then fires more nerve impulses. And it's this area which is affected by drugs a lot of the time. So depressant drugs might slow this transmission, this diffusion down, or release of neurotransmitter, and stimulator drugs might increase the amount of adrenaline or noradrenaline released. So one thing that nerves do, which we need to know in detail, is control reflex actions. Reflex actions are rapid and automatic responses that don't involve conscious thought, although you can override them if needed. And they are rapid because that protects your body from danger. So burning yourself, for example, if you pull your hand away quickly, you're less likely to get serious damage. You need to learn all the parts of this thing called the reflex arc, which shows you what happens in a nerve impulse. So effectively what happens is you have a stimulus, which is normally a level of energy change. So let's say you put your hand on a hot plate. The stimulus will then be detected by something called receptor cells, which will be the pain receptors in your skin, in your hand. That then triggers an electrical impulse, which goes down your sensory neuron, and you need to know the order of these neurons. That would then jump over a synapse to go into the relay neuron, which is in your spine and your central nervous system. Your central nervous system makes up your spine and brain. Over a synapse then to the motor neuron, and then the motor neuron would then tell an effector to respond. An effector is always either a muscle, which can contract, or a gland, which would release a hormone. In this case, it's a hot plate, which you picked up, so your response would be to drop the hot plate, so it would be a muscle with the effector you're using. Now, your brain isn't involved, but your brain can get involved and override, or it is told momentarily later. So that brain is often called the coordinator in reflex actions. Now, just to show you an example of this and why this is so important to learn in exams, recently this has been a 6-mark question here where it just shows you that there's neuron A, B and C, and you're told the word receptor and you're told the word effector, but the question is describe how they coordinate a reflex action. And the mark scheme here you can see, you're expected to know the receptor detects the response. Because of the example they give you, they talk about, you don't have to, because it's not specific, you don't have to give, you can just give any example. It talks about electrical impulses, which you need to know that the receptor instigates. Then that travels down neuron A. You need to name neuron A. You then need to talk somewhere about synapses and chemicals crossing the synapse. Then go through B, which you need to name as the relay neuron, and the motor neuron, neuron C. You then need to say that the effector causes the response, and that can be a muscle or a gland. So it shows you the importance of knowing that in detail. Now, you should have done a required practical. I'm not going to go over this required practical much. You need to look at your own notes on this really, but the ruler practical, where you might have hopefully had a partner, you dropped a ruler and you tried to catch it. Now, you might have done this before and after drinking a caffeine drink. You might have done it with your right and left hand to look at the differences between it. But AQA said that you should use your weaker hand for this experiment. They said you should sit down and look ahead. You should then place your arm on the edge of the table so it's in the same position. Your partner holds the ruler just between your thumb and finger, and then without warning they let go and you catch it as quick as you can, and you record that in the table and you can convert it into actual times, and you can practice maybe your reactions before or after caffeine, for example. Now, you need to know about the brain, and the brain is a complex organ which we don't study much at GCSE because it is so complex. It's actually really difficult to study. And the brain is just basically millions of neurons that are all interconnected. So all the different nerve impulses give us our memory and thoughts. And there are 3 sections of the brain you need to know about. So the first section, highlighted in purple, is the cerebrum or the cerebral cortex, and that is involved in your consciousness, your intelligence, your memory and your language. So I always think of this as, this is what I think of the brain as, this outer layer, this sort of pink layer, and I always think in my head that brain gives us our intelligence and our sort of personality. You've then got the cerebellum at the back, and your cerebellum is controlled of your muscular activity. And then you have the medulla. The medulla means middle in biology a lot, so there's a medulla in the kidney as well. And your medulla is involved in unconscious activities such as heartbeat and things like that. So your medulla is quite an important thing there. Now, studying the brain is difficult because your brain is such a complex organ that if you start messing with it, you're likely to cause some serious health issues and affect people's behaviour. So we can only really study the brain using people that have had brain damage and compare them to other people that haven't. We can electrically stimulate parts of the brain and see what effect that has, or we can use MRI scans where people get their brain activity gets monitored as they complete tasks such as crosswords, etc. And the brain is difficult to study because of that problem of it can be damaged, and even trying to treat the brain with drugs is difficult because a lot of the time the brain won't allow drugs through the membrane. So if you're doing separate science, you need to know about the structure of the eye, and you need to know effectively the parts of the eye and the key parts of the eye. So the outer part of the eye is something called the cornea, and the cornea's job is it protects the eye, but also as light enters the eye, the cornea starts the refraction process. It starts bending the light towards the back of the eye, which is important. You then pass through the hole, and that's all the pupil is, and it is a hole that allows light to go through, and then the light passes through the lens, which continues to bend the light through to the back of the eye, which is what detects the light. And the back of the eye is called the retina, and those are the receptor cells that detect the stimulus. Other parts of the eye that you need to know about are ciliary muscles, which we'll talk about shortly, and suspensory ligaments that are important in the eye. Probably also worth knowing, this bit here is called the optic nerve, and that's how the brain and eye are connected. Instead, you need to know what these parts do, what they do. So different things that are changed. The first thing is what changes size to make sure the correct amount of light enters the eye. So that is the pupil, which is affected by something called the iris, which are muscles that control the size of the pupil. And the pupil gets smaller in bright light, and it gets larger in dim light to allow the correct amount of light to enter the eye. You then have the ciliary muscles, and we're going to talk about those in a moment. They contract to change the size of the lens and pull on the suspensory ligaments, and the lens itself refracts the light so you can focus and see things clearly. So there's something called accommodation, which means how do our eyes change to look at distant objects and look at far objects. So we start off with distance objects. So objects that are close to us. You need to know these key things. So the ciliary muscles here relax. Okay, now when the ciliary muscles relax, the ciliary muscles being this label here, what that means is they stop pushing inwards, and it causes the suspensory ligaments, which are here, basically to be pulled tight. So when the ciliary muscles relax, the eye effectively pulls outwards. That pulls the suspensory ligaments tight. The lens is pulled thin, as you can see in this picture here, and therefore that means the light reflects less and it hits the back of your eye as needed. That's where the light rays cross over. For near objects, the ciliary muscles contract, and what that does is it pulls the eye inwards at this point here. That means the suspensory ligaments are not pulled, the lens gets fatter and rounder, and therefore the light then hits the back of the eye so you can still see clear objects. And you need to learn those points, particularly for higher tier, in separate science. Now there are two conditions you need to be aware of, and briefly how we correct them. So there is something called myopia, which is short-sightedness. And I always use the word short here constantly to remind myself what the problem is here. So this is when you can see objects close to you. So literally you are short-sighted because you see closer objects to you, but you can't see distant objects. If uncorrected, the image forms in front of the retina. So the light crosses before the retina. So I always think of it as crossing short of the retina. And why would this happen? Well, think about it logically. It could be because the eye is too big, or your lens is too powerful, so the light's crossing too early. And the correction of this is a diverging lens. So what you'd have is something like this, which just pushes the light rays out a bit further so they don't cross till later on until they hit the retina. And the opposite of that is hyperopia, which is long-sightedness. And again, if you think of the word long here, this helps you understand it. You can see things in the distance clear, longer objects clear. And the reason that it happens is because the light doesn't cross over, the light rays don't cross and meet or converge until long of the retina, sort of behind the retina. Now, why would this happen? Because your eyeball might be too short, and people, when they're born, sometimes have long-sightedness because their eyes are too small, but then they grow, and young children who often have this will grow out of having long-sightedness. It could be the lens is too weak, however, and it could prevent focus. And correction for this could be that you would use a converging lens as seen in the second diagram here, which just makes the light rays cross over sooner on the back of the retina, forming the image correctly for you.